Online lecture covers Chapter 11 on the nervous system. This is part 2 of our nervous system lecture series covering neurophysiology. In order for us to dive into neurophysiology, we first need to define a couple of terms. So the first term is voltage. Voltage is defined as the measure of potential energy generated by separated electrical charges. So voltage is the measure of potential energy generated by separated electrical charges. Another way to describe voltage is to just say what is the charge in that location. So the voltage really refers to the charge in a specific location. The potential, or the potential difference, is then going to be the difference in voltage at two locations. So the potential is the voltage measured between 2 points, or the charge difference between two locations. Current is defined as the flow of electrical charges. So charged molecules are known as ions. Ions are going to move according to their chemical gradient, so their electrochemical gradient. So in other words, they're going to move from an area of high concentration to an area of low concentration. So they're going to move based on the concentration difference. So they're going to move from areas of high concentration to areas of low concentration, or down their concentration gradient. Or they're going to move according to electrical charges. So positively charged ions want to be by negatively charged ions, and vice versa. So ions will also move towards ions of an opposite charge. The last term is resistance. Resistance is defined as the hindrance to charge flow. So anything that prevents those ions or those charged molecules from moving is providing some resistance to charge flow. In general, the greater the voltage, the greater the current. The greater the resistance, the smaller the current. So in other words, the greater the difference between charges in two locations, the greater the current, or the greater the flow of those electrical ions. The greater the resistance, or the greater the hindrance to charge flow, the smaller the current, or the smaller the flow of electrical charges. So neurons are going to function by rapidly changing the voltage across their plasma membrane. So neurons function by rapidly changing the voltage across their plasma membrane. The difference in voltage across the plasma membrane is known as the membrane potential. So potential is the voltage measured between 2 points. So the membrane potential is going to be the difference in charge across the plasma membrane. So neurons are going to function by rapidly changing the voltage across that plasma membrane, or rapidly changing their membrane potential. In doing so, they're sending and receiving information in order to perform the overall function of the nervous system, which is to control. So neurons can rapidly change their resting membrane potential. When they do so, this is called a nerve impulse, or an action potential. So when neurons rapidly change their resting membrane potential, they're generating a nerve impulse or an action potential, which is the way in which neurons communicate with one another. When a neuron sends an action potential to another neuron, the point at which those two neurons are communicating is known as chemical transmission, or a chemical transmission site, which is known as a synapse. So when one neuron passes an action potential to another neuron, and then they're communicating with one another, that's known as chemical transmission or a synapse. So neurons function to communicate with one another or to send and receive information by rapidly changing their resting membrane potential, or their RMP. All cells, not just neurons, have membrane potentials. So all of our cells actually have a difference in charge across the plasma membrane. However, not all of our cells can change that membrane potential. And so we refer to the membrane potential of a neuron that is not communicating as the resting membrane potential. So when a neuron is not sending or receiving electrical impulses or nerve impulses, the membrane potential is known as the resting membrane potential. We would call the membrane potential just the membrane potential in any other cell that doesn't rapidly change their membrane potential, because it's always about the same. So they don't change the voltage across that plasma membrane as a means of communicating. So neurons, as well as muscle cells, are what are called excitable cells, which means that they're capable of rapidly changing that membrane potential as a means of communicating with one another. So neurons and muscle cells are excitable cells, which means they have a resting membrane potential that they can rapidly change in order to communicate with other cells. In general, the membrane potentials of our cells is going to be anywhere from -50 to about -100 millivolts. So the resting membrane potential is also known as the potential difference that exists across the membrane. We measure it in millivolts. In neurons, the resting membrane potential is going to be -70 millivolts. So in neurons, that resting membrane potential is about -70. That means that when you measure that resting membrane potential right inside of the plasma membrane of a neuron, it's going to be pretty negative. So it's going to be -70 millivolts. So the inside of the cell is more negative than the outside of the cell. We say then that the membrane is polarized, which just means that there's a clear charge difference across that plasma membrane. So one side of the plasma membrane is more negatively charged, the other side of the plasma membrane is more positively charged. So that just is how we would define if a membrane is polarized. So if it's polarized, either side of that plasma membrane has a difference in charge. It's important to realize that this electrical difference really only exists at the plasma membrane. So at the plasma membrane, we see a difference in charges or a difference in voltage. However, in reality, if you look at the overall cell and the overall extracellular fluid, the inside of the cell and the outside of the cell are actually electrically neutral. So the inside of the cell and the outside of the cell are actually electrically neutral, but when we look close to that plasma membrane, we do see a difference in charge across that plasma membrane, which generates that resting membrane potential. There's going to be a difference in charge across the plasma membrane because of differences in ion concentrations inside and outside of the cell, and because the membranes have different permeabilities to different ions. So the resting membrane potential, or this difference in charge or voltage across the plasma membrane, exists because there are differences in ion concentrations inside and outside of the cell, and because the membrane has different permeabilities to these different ions. So first we're going to take a look at differences in our ion concentrations that exist inside and outside of the cell. So inside of our cells, we see that we have a high concentration of potassium, and we have a high concentration of these protein anions. So inside of the cell, we're going to have a high concentration of potassium and a high concentration of protein anions. Outside of the cell, so in our extracellular fluid, we're going to have a high concentration of sodium ions and a high concentration of chloride ions. So we have more sodium outside of the cell than inside of the cell. We have more potassium inside of the cell compared to outside of the cell. Therefore, there's a concentration gradient for potassium to want to leave the cell or diffuse out of the cell, and there's a concentration gradient for sodium to want to enter the cell. So because of these ion concentration differences, sodium wants to enter the cell and potassium wants to leave. So the resting membrane potential is caused not only by differences in ion concentrations, but also due to differences in membrane permeability to these ions. So if a membrane is permeable to some ion, that means there's some type of a channel or a transporter that allows that ion to move across the plasma membrane. So if we say that a membrane is permeable to some ion, we just mean that there's some type of a channel or transport protein that's allowing that ion to cross the plasma membrane. So we're going to look at ion channels. So in our neurons, we're going to have ion channels that are going to allow for that neuron plasma membrane to be permeable to potassium and permeable to sodium. Ion channels could be considered leakage channels or non-gated channels, or gated channels. A leakage channel is always open. So in a leakage channel, ions are always free to move through that channel down their concentration gradients. Ion channels could also be gated. They could be chemically gated, voltage gated, or mechanically gated. In a chemically gated channel, some chemical or some ligand must bind to that channel, causing it to open or close. In a mechanically gated channel, some mechanical stimulus must occur in order to open or close that channel. A voltage-gated channel is going to open in response to some change in voltage, so some change in charge. So like I said, our neuron plasma membrane is going to be permeable to sodium and potassium ions. Sodium and potassium ions are going to be allowed to cross the plasma membrane through leakage channels. So first we'll start with our potassium leakage channels. So a neuron's plasma membrane has potassium leakage channels. Because of the difference in ion concentrations across the plasma membrane, we have high potassium inside of the cell, low potassium outside of the cell. That means that if you have these leakage channels present in the plasma membrane, potassium is going to want to leave the cell and enter that extracellular fluid. So because you've got a bunch of potassium inside of the cell, not a lot outside of the cell, potassium is going to be continuously leaking out of that neuron and entering that extracellular fluid. If you're losing a ton of positive ions, That's then going to make your resting membrane potential very negative. So if we exclusively had these potassium leakage channels present, a bunch of potassium is going to leave, so therefore the resting membrane potential ends up becoming extremely negative, or the inside of the cell ends up looking very, very negative. So we would reach a resting membrane potential of about negative 90 millivolts if we exclusively had potassium leakage channels. In reality, we also have leakage channels for sodium. So sodium's concentration is higher outside of the cell compared to inside of the cell. So therefore, there's a concentration gradient for sodium to want to enter the cell. So we have sodium leakage channels in the neuron plasma membrane. These sodium leakage channels are going to allow for sodium to leave the extracellular fluid and enter the cell. So they're going to leak into our cells. As sodium is leaking into our cells, that's going to bring our resting membrane potential down to about negative 70 millivolts. Or I guess back up, I guess you could say, to negative 70 millivolts. So why don't you think we see a bigger change in that resting membrane potential when we add in our sodium leakage channels? Well, if you look at this diagram, what it's showing you is that we have more potassium leakage channels than sodium leakage channels. This is a simplified diagram that's showing you essentially a 2-to-1 ratio of potassium leakage channels to sodium leakage channels. In reality, it's actually a 25-to-1 ratio. So in other words, the membrane is 25 times more permeable to potassium than to sodium. So what that means is there's 25 times more potassium leakage channels than sodium leakage channels. So that means we're going to lose a lot more positive ions as potassium's leaking out of all of those channels in comparison to all the positive ions that we're going to gain through those few sodium leakage channels. So the membrane is 25 times more permeable to potassium than to sodium, which means that we're going to lose a lot more positive ions than we're going to gain. So that produces a resting membrane potential of about negative 70 millivolts. The issue here is that if we were to just have leakage channels present in our plasma membrane and that was it, eventually ions are going to stop moving once they reach equilibrium. So potassium is going to continue to leave the cell, sodium is going to continue to enter, all the way until we've got equal concentrations of ions on either side of the plasma membrane. So this two-way diffusion will eventually cause the gradients to disappear. So therefore, that resting membrane potential or that difference in charge is also going to disappear. So this is part of what happens at death, is that your neurons are no longer going to function because we no longer have the difference in concentration across that plasma membrane for sodium and potassium. So what's going to keep those ionic gradients in a steady state? So in other words, how am I going to keep potassium high inside of the cell and keep sodium high outside of the cell? The sodium-potassium pump is going to be our key player for keeping those ionic gradients in a steady state. So we've seen the sodium-potassium pump before. This was our example for primary active transport. So in the sodium-potassium pump, we are going to have this membrane protein here, looks like Pac-Man. When it's open to the inside of the cell, we're going to have 3 sodiums come in and bind to this transporter. When the sodiums bind to the transporter, it's going to cause the hydrolysis of ATP. So in other words, this transporter is going to become phosphorylated. So it's going to harness the energy associated with the hydrolysis of ATP in order to change its conformation and open to the extracellular fluid. The sodium ions are then able to be released or expelled to outside of the cell. Once sodium ions are released outside of the cell, 2 potassium ions are going to come in, and they're going to bind to this pump. When those potassium ions bind to the pump, that triggers the release of this phosphate, and that triggers this membrane protein or this transport pump to regain its original conformation. So it's now going to be converted back into its original conformation, which was open to the inside of the cell. So now those potassiums are going to leave and they're going to enter the inside of our cell. Another ATP can then come in and bind, and then this process starts over again. So our sodium-potassium pump is going to come back again in the nervous system in a really big way. So it's extremely, extremely important that you know these steps of our sodium-potassium pump once again. So in general, our sodium-potassium pump is pumping sodiums from inside of the cell outside of the cell. So it's pumping 3 sodiums out of the cell, and it's pumping 2 potassiums inside of the cell. So this is then going to compensate for those leaking ions, and it's going to keep the concentration of potassium high inside of the cell, and it's going to keep the concentration of sodium high outside of the cell. So the sodium-potassium pump is going to be our key player in helping to maintain those ionic gradients so that potassium and sodium can be continuously leaking out of our cells or leaking into our cells. So one last thing I want to mention is that because our membrane is 25 times more permeable to sodium than potassium, that really negative resting membrane potential is going to be mostly due to the leakage of our potassiums or the potassiums diffusing from inside of the cell to outside of the cell. So our resting membrane potential is very negative, predominantly due to the loss of all of those potassium ions. So now we're going to watch a video on generating our resting membrane potential.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on 2 factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.